It's my great pleasure to introduce our second keynote speaker, Dr. Atul Chopra is an esteemed physician scientist with dual appointments at CASE and UH. Dr. Chopra's groundbreaking research spans genetics, rare metabolic diseases, and novel hormone therapies, contributing significantly to precision medicine. With a background that bridges basic science and clinical care, his work has expanded our understanding of metabolic disorders and also opened new therapeutic avenues. Today, he'll share insights from his latest research, further highlighting the intersection of genetics and innovative treatments. Please join me in welcoming Dr. Atul Chopra.
Thank you for that very kind introduction. It's a real pleasure to be here. I think I presented at this conference several years ago. So I've got a few updates. Okay. I'm a medical geneticist. So our stories begin with humans. And really what I want to do is tell you a story today. The theme of the story is how you can go from one patient and make biological observations, just one patient, to start the whole journey off. And that's enough to open up a whole Pandora's box of discoveries. I won't talk about drug development today, just basic discovery. And really it's all about how do you discover new hormones. Okay, so several years ago, maybe like 12, I was still a resident at Texas Children's. I saw her in the clinic. And her name is Lizzie, and I'm not breaking HIPAA because this is all in the public domain. And the reason I saw her was for her extreme thinness, a BMI of about 10. You don't really see this all that often. Just looking at her phenotype, you can kind of tell you don't see this phenotype just walking around. It's very rare. And that's kind of all we knew about it. It had this weird, obscure kind of name, neonatal progeroid syndrome, which has since changed into Marfanoid progeroid lipodystrophy. Mouthful. It doesn't really matter. The reason I saw her is because of her extreme thinness. I was like, let's try and figure this out. That's why medical genetics is so great. This is not really a pitch for you guys to go into residency in medical genetics, although that would be great. But we solve mysteries, and it's fun. It's a ton of fun. You see a patient like this, and it opens up this whole thing about what's happening in there, right? Okay. Even in the rare disease world, patients tend to know each other, especially in the social media age. Lizzie knew Abby, who also happened to live in Texas. There's been maybe 10 patients discovered with this syndrome. So the fact that there were two in Texas and they were both willing to come to Texas Children's and see me was a minor miracle in and of itself. But they were looking for answers too. Okay, so Abby, BMI of 13. Let's just stick with the thinness. There's a bunch of other things happening that I won't get into. I want to tell a clear story. Okay, so thinness. When you see patients with thinness, in the clinic here, I direct a clinic specifically devoted to body weight. I see kids with extreme obesities, mostly. Extreme thinness, extreme obesities. The question remains the same. What does the energy balance equation look like? Because your body weight, all creatures on Earth, our body weights are determined by how much energy goes in and how much energy is lost every day. That's the energy balance equation. So in the context of extreme thinness, Lizzie, who has a BMI of about 10, eats about 955, say 1,000 kilocalories a day. That's about half to 2/3 of an age-matched, sex-matched control. Low caloric intake, okay? The second patient, unrelated to her, also eats about 2/3 of the calories of an age-matched, sex-matched control. All right, so now the story is heating up. This kind of doesn't happen by chance. And this is not a psychological disorder. They have been thin since birth, okay? Big distinction there. So we're not looking at anorexia. This is not a psychological disorder. There was something else happening inside. The energy balance, when you look at numbers, they kind of tell you the truth. And what you're trying to get at is, where does the fault lie? Where's the problem? What's broken down? Is it energy intake or is it too much energy expenditure? Both could conceivably cause thinness. Well, energy expenditure is also low. Both of these numbers, energy expenditure numbers, are low. So you have thinness You have low caloric intake, low desire for food, and low energy expenditure. Well, no, low energy expenditure could never account for thinness. If anything, it should give you more body weight, right? Everyone with me so far? So the culprit here, very clearly, you don't really, not rocket science, you don't really have to know much more. The culprit here is low appetite, okay? Fact. Okay, 2 unrelated patients in the world with this weird form of extremely rare thinness that don't like food that much. They don't desire food as much as the rest of us do. The second thing to look at when you see a patient with extreme thinness, we call them lipodystrophies. I'm not going to use very big words today, but lipodystrophy is basically disordered fat. It's something you have to rule out. It's in the differential diagnosis. Okay? So it's like fat that doesn't develop well. When we see patients with bona fide lipodystrophies, what you find is diabetes, usually, insulin resistance, because fat is necessary for glucose to disappear into under the direction of insulin. All right? So when fat doesn't develop well, all that glucose stays outside and you get diabetes. These patients have the opposite. They have insulin sensitivity, low insulin, and that's enough to do the job. That's insulin sensitivity. Glucose is normal, insulin is low, all right? So this is a weird result to get for a patient with an extreme thinness. Again, this was telling us something. And both patients in the clinic, when I'm asking them questions about, hey, do you drink a lot of water? Do you pee a lot? These are diabetes-related questions. The answer to all of that from 2 unrelated patients was no, we don't have any of that. What we do have is we wake up in the morning and feel sweaty and feel a sense of impending pass out and feel just not great. And all of that is alleviated by drinking some Gatorade or eating a chip or a cookie, high glycemic food. Okay, that smacks both 2 unrelated patients. I keep referring to the fact that they're unrelated because 2 people telling you the same story, that it's happening by chance goes down, all right? So they have neuroglycopenia, less glucose, low insulin, a weird result, okay, for somebody with this kind of thinness. So what's going on? Not knowing anything about this disorder, and at Texas Children's, we had a huge medical genetics practice, 25 active clinical geneticists, all right? And of course, we did conference after clinics, and none of them had seen this disorder. None of them could offer any insight. What do we do in my practice when we don't know what's going on? We sequence them, right? So it's like throw the whole kitchen sink at them and see what comes back. Sequence the whole genome, the exome in this case. And what came back was also a very weird result. Mutations in 2 unrelated patients again, in FBN1, the Marfan syndrome gene, and both had the same character of mutation. They were truncating mutations at one extreme end of the gene. We call it the 3' end, all right? 5', 3'. On the right end of the gene, they were truncating mutations that were chopping the gene, all right? So whatever was produced would be chopped up, basically reduced in length. Okay, so already this is looking like, hey, this is real. This has to be real because the fact that they're 2 different patients unrelated to each other, have the same phenotype, and have mutations in the same gene with the same character. This already doesn't happen by chance. But now FBN1 was on my radar. So now one can do a literature search. And out come 5 additional case reports of others around the globe making the same association. Okay, patients with extreme thinness with 3' truncating mutations in FBN1. Now it is definitely not by chance. 7 unrelated patients in the world, same thing, it's not by chance. I guess we have to use big words. In our language, we just have to use big words. This would be called a tight genotype-phenotype correlation. And it's sort of the gold standard that all geneticists live and die by. That's what you're trying to get to, a tight genotype-phenotype correlation. So you're looking at the right cause, molecular cause, for whatever you're finding on the surface. Okay, so what's happening? This is the protein product. It's called profibrillin of FBN1, okay? Profibrillin is cut normally in all of us, normal stuff. It's cut by a protease called furin at the C-terminal end, at one end, and that makes profibrillin into fibrillin. This big green protein is fibrillin. It's an extracellular matrix protein. It's huge. All right, it makes basement membrane. It makes structure of cells. It's an extracellular matrix protein, massive protein. Well, every cleavage event yields two proteins, right? So here's this little guy, only 140 amino acids. All this was already worked out by others who were studying Marfan syndrome, FBN1, fibrillin. This little guy is also released, but no one had ever studied the second protein before. They had studied fibrillin, they had studied this cleavage event as a means of trying to understand how profibrillin becomes fibrillin, very basic science, and how extracellular matrix forms. But nobody had ever found any cause to study this little protein before. What's happening in our patients? A bunch of gibberish, but really what this is here's our patients, here's my patients, and here's 5 other cases, right? And what we're looking at is the last 2 lines, the C-terminal most lines of the amino acid sequence, of the protein sequence. What's happening is this little guy, this RGR, KRR motif is where the cleavage happens. The scissor comes along and cuts. Everything north of that is fibrillin, and everything south of that is the second protein. And the second protein is deleted in all the patients. It's gone. Remember, I said these are truncating mutations. Well, the effect of the truncation is the second protein is gone. It's just deleted. So from that emerged a very simple, again, not rocket science, very simple hypothesis that turned out to be correct over time. The second protein is an independent functional entity, and its reduction in some mysterious way that we hadn't understood yet, we have understood now, causes this phenotype. All right? Everyone with me? Okay, how do you go about looking at that hypothesis? How do you go about examining? The beauty of a hypothesis is now you can do experiments and actually test whether that statement, remember I made the statement here, whether this statement is correct. The first thing to do, if it's an independent functional entity, that it must actually survive. It must, after the cleavage, it's not just taken out by the body and gotten rid of. It actually lives, okay? So does it exist? And we found that it exists in the blood. So these are 6 basically unaffected control subjects, right? Control subjects is you and I. And it's present in our blood at this 30-ish kilodalton size. If you use bacterial cells to make it, it runs at 16 kilodaltons, about half the size. And that's because bacteria can do post-translational modifications. They can add on a bunch of things to proteins. But in humans, it's present in our blood. And it lives at 30 kilodaltons, not very important. But it exists. That's the point of this. That's step number 1 in trying to prove this hypothesis. So we named the second protein asparagine. Funny story here. After we published our first paper, and we named this asparagine because patients have a white adipose tissue deficiency. I've just shown you patients with like an adipose tissue deficiency, fat deficiency, so white. And asparose is the Greek word for white, and asparagine is also highly expressed, FBN1 is highly expressed in adipose tissue, white adipose tissue, so asparagine, for better or for worse. So the first paper comes out in 2016, and I get a call from this reporter at the Times of London. Okay, I'm sitting in my office at Baylor. And he's like, I'm a classicist, I'm a Greek classicist, and I can't find this word, asparose. And all diligence I'd done was Wikipedia. Okay, what's white for, or the internet, what's white in Greek? And asparose came back. And a colleague, Nicholas Mitsiadis, who's Greek, I was like, hey, is this right? And he was like, yeah. And now this classicist, okay, is telling me that this word doesn't exist. He's never heard it. So for me, it was an oh crap moment. Like, these guys have named a protein after some gibberish word that doesn't actually exist. And it's in print by this time, all right? I can't take it back. And then he goes away and he comes back 30 minutes later. He calls me again 30 minutes later and is like, I found it. It's in modern Greek. It's like, I don't care if it's in modern Greek, as long as it's in some Greek out there. So that was an few moment. Yeah, that was great. Okay, so asparagine. I'm going to refer to that second protein as asparagine from now on. Remember I mentioned the patients have this, they report at least, this feeling of hypoglycemia, right? That smacks of hypoglycemia. They also have lowish insulin levels to maintain glucose levels being normal, and I couldn't really fast them. They're very intolerant of a fast. They have to eat every 15, 20 minutes a high glycemic index food to keep from feeling bad, that feeling of hypoglycemia. And so I couldn't really fast them very well overnight. And really, I think that's why we missed the hypoglycemia in the clinic to begin with, but we did catch the lowish insulin. Insulin is a good readout for glucose. If your glucose is low, insulin will buffer and prevent. Glucose is essential, life-giving, okay? Neurons burn glucose constantly, and without glucose, we will seize and die, all right? So the body, and obviously we can't eat all the time. So the way the body combats that is when food isn't available, your liver makes glucose, but it's not making it at the level of like drinking sugar water. So insulin levels go down to keep glucose outside in the bloodstream so that neurons have constant access to glucose. So low insulin is actually a readout for lowish glucose, is how we've understood it over time. But really, what does asparagine do? I did a bajillion experiments, okay? But the ones these are the key ones from that first story. You do something called a glucose clamp or hyperinsulinemic euglycemic clamp, and that tells you the level of glucose production by the liver. Okay, it's the gold standard test. When you have too much asparagine, and we can do that in a couple different ways, the hepatic glucose production is elevated. And when you have too little asparagine, we can do that in a genetic way being done here. I won't get into the details of that. You have too little hepatic glucose production. Pretty good. It was good enough to know that, this is real. When you throw glucose into the body of a mouse, if you just inject, asparagine into the body of a mouse, you just inject it, near instantaneous, you get a spike in glucose because the liver starts pumping it out. Okay, so that's the first function of asparagine. So we published this paper in 2016, and the title was Asparagine is a glucogenic protein hormone. But the story is just beginning. This was the discovery of asparagine and the first physiological task that it accomplishes. But hormones generally do many, many, many things. So the story actually continues from there. And of course, by this time, in the chronological order, we haven't actually solved the mystery of why the patients are so thin. The second story gets into that. Remember I mentioned that the patients are a loss of function model. They have too little asparagine. Think of the patients as they're a deficiency model of asparagine. When we put the same mutation in a mouse, the mouse has that exact same phenotype and has barely any asparagine that we can detect in its blood, okay? And of course, we've deleted asparagine, right? This was a natural experiment. Nature deleted asparagine in these patients, and they have this phenotype of thinness. We found that asparagine is not only present in the blood, as I've just shown you, in that Western blot, it's also present in the CSF. And in the CSF and in the blood, which I'm not showing, it's fasting induced. So you have lack of food or you don't eat, asparagine levels go up in the CSF. So it's present in the blood. It's present in the brain. Not only is it present in the brain, plasma asparagine can cross the blood-brain barrier. Super important into some of the therapeutics that we are developing now, which I really won't get into unless you're curious. This is an IV injection in rats, an IV injection of asparagine. And an hour later, boatloads of asparagine have shown up in the CSF. And that only happens. This protein is too big to just diffuse across. It's about 30 kilodaltons. It's past the threshold for stuff that diffuses across. So this is some sort of active process, which we haven't really looked into anymore. But anyway, asparagine crosses the blood-brain barrier. And this gets to the crux of why the patients are so thin and have low desire for food, because asparagine does the opposite. When you don't have asparagine, you have low desire for food, low appetite. And when you give a mouse too much, an injection of asparagine, the mice eat like gangbusters. So they eat a lot more. 5 grams, 6 grams of food a day for a mouse is a lot of food. Okay? So we can basically control this process at will. You increase asparagine, food intake goes up, glucose goes up. You get rid of asparagine. You can do that genetically in humans, or nature does that genetically in humans. You can do that in mice. We've put the same mutation in a mouse. You can do that using monoclonal antibodies against asparagine. All the methods basically show the same thing. You get rid of asparagine or reduce its levels in the blood, you get a reduction in appetite, thinness, and reduction in glucose insulin levels. How does it stimulate appetite? It works on these neurons. Research is very reductive. So we only know what we know, and we don't know what we don't know, which sounds like a stupid, it just sounds like an obvious statement to make, but it's fairly profound for basic science. We don't know if this is the only way asparagine increases appetite, but it's one of the ways. Now, these neurons are pretty well known, AGRP neurons. They're in the hypothalamus, a nucleus called the arcuate nucleus, and they control appetite. So when these neurons fire, The mice become hungrier and go to the food and start eating. Okay, people had shown that many, many years, decades before we came along. We throw asparagine on these neurons and they start firing. The resting membrane potential goes up, that's this red line, so they depolarize, in other words. And their firing frequency, it all bunches up, so they start firing more frequently. So that's one mechanism by which asparagine increases appetite. We've done a whole bunch of work on this. And this story we finished about 18 months after that first story. Asparagine is a centrally acting orexigenic hormone. Orexigenic is appetite stimulating. Okay, by this time we had been, our lab had been recruited here, and I'll tell you a funny story about that at the end. What is the identity of the asparagine receptor? You have a hormone, it needs a receptor. No hormones work without a receptor. So a hormone is floating around in the blood and the CSF. It makes contact with cells. It can't talk to that cell and activate it without another protein that it needs to touch on the cell surface. That's a receptor, okay? So we started looking for it in the liver and failed. For like 3 years or 4 years, we failed. It just didn't come up in the kind of experiment that I'm about to describe. So we were like, okay, let's do it in the brain. So we did this experiment in the brain, and it's a fairly simple, I think you'll find that research is, you increase things and you decrease things and you look for observation, like, hey, what happens? You look for correlates of that. In this case, it's also a very simple experiment. You take a bunch of mouse brain, you mash it up in 2 bowls. In one bowl, you throw asparagine, okay, on top of it, recombinant asparagine. The other one gets an irrelevant protein, GFP, not found in humans or mice, right? And you pull this asparagine with antibodies. And you ask the question, what else comes along? And then you ask that question using a technique called mass spectrometry. So fairly simple in concept. You pull it and ask what other proteins come along with it. All right? So we did this experiment 3 times and found 58 proteins that came with asparagine each of those 3 experiments. We actually did it 4 times. 4th time failed. But this was good enough. Out of the 58, there was just 1 receptor. And it was a big surprise that it was this receptor, which I'd never heard of before. It's called protein tyrosine phosphatase receptor delta. Another mouthful, but it's a phosphatase. It's an enzymatic receptor. So it has this stuff hanging out outside the cell, just flapping in the wind. That's where asparagine binds. And then it has a single transmembrane domain. So that's where it crosses the membrane. And then an enzyme, actually 2 enzymes inside, okay? And they're both phosphatases. They take phosphate groups off its substrate, whatever the substrate may be. So, okay, we'd never heard of this guy before. So at this point, you start looking for patterns. And I'll show you the data, of course. But the pattern that you're looking for is, if you get rid of this guy, do you get the same disease as the lack of asparagine? Do you get the same phenotype as lack of asparagine? So again, not rocket science, super simple sort of level of thinking, just very linear. Somebody else had already done this experiment. Okay, this is our data. But way back, a Japanese group like 30 years ago had made this knockout mouse for this protein, right? I was 5 years old. And what they found was these mice did not like to eat very much, had a bunch of other things, but did not like to eat very much and weighed less. They were thin. So we got the mice from Japan, and we found that all that was correct. The mice weigh a lot less, and they also don't like food. Not as much as wild-type mice that have this protein. So mice without PTPRD, asparagine receptor, that came up when we pulled asparagine and asked what else comes along with it from neurons, mice without PTPRD look and behave like mice without asparagine. So this was important. Okay, this level of correlation was really important. If these mice didn't have this phenotype, we'd kind of know something is off with the story. A negative experiment doesn't tell you all that much. There are other ways in which this could be right and still not show the phenotype. But you do have to get lucky, okay? So luck is a very important component of making discoveries. And this was getting lucky in a big way. Number 1, we found it in an unbiased way. And number 2, the mice without it look and behave like mice without asparagine. So we kind of knew this was looking promising, that we are on the right track here. But the upshot experiment The really important experiment is when you get rid of this guy, do mice still respond to asparagine? Everyone can see why that would be the clear experiment to prove that this thing is necessary for asparagine to work. Everyone with me so far? Right? So we got rid of it, and we gave the mice too much asparagine. And I won't get into how we're doing it, but these mice now, the ones where so plus is this receptor is still there. Okay, both alleles of it, both copies of it are still there. So this is still plus here, it's just cut off. So these are wild type mice on this side, okay? On this side are PTPRD knockout mice. In both cases, we're giving one group of mice too much asparagine. And in wild type mice, of course, we already kind of knew this, that when you give them too much asparagine, they eat more. So this is food intake. That does not happen in knockout mice. I'm just showing you one snapshot. We've done this experiment a bunch of different ways. So this was sort of the critical experiment. Of course, we had a bunch of mechanism to do, and what are the substrates, and what does it take the phosphate group off of, and when PTPRD is not there, do AGRP neurons still respond to asparagine, all that. So the paper is chock full of data, right? I'm just showing you snapshots. And we published this in 2022. So protein tyrosine phosphatase receptor delta serves as the orexigenic asparagine receptor. A different group by this time had published what the receptor is in the liver, and it turns out it's a GPCR, unrelated to this enzymatic receptor. So this experiment that I just showed you, when you look at glucose levels instead of looking at food intake, the glucose levels go up even when PTPRD is not there, telling us that PTPRD is not essential for asparagine's glucogenic effect that comes through the liver, but it is essential, completely crucial for asparagine's orexigenic effect, which happens in the brain. So that's why the title of the paper, it's the orexigenic asparagine receptor. The glucogenic one is different. And there's precedence out there for a hormone to use multiple receptor types. Okay, this also seemed a little weird, and I got a bunch of questions at conferences about it, but we found a few papers with showing that different hormones like estrogen, insulin even works with different kinds of receptors. Bile acids, a different kind of hormone, works with a nuclear receptor and a GPCR. Hormones tend to do this. We do not understand the structural basis of this, but that's a question for tomorrow. Okay, orexigenic asparagine receptor. Now it opens up a whole bunch of opportunities. One thing you can do is ask the question, where is the asparagine receptor expressed? What tissues of the body have too much of it, have a lot of it? And the tissue that kind of stands out is the cerebellum, this stuff here, right? This is in humans, by the way, RNA-seq in humans. And the cerebellum has a whole bunch of the asparagine receptor. And of course, I've just told you that asparagine works in the hypothalamus. So the question became, what is the asparagine receptor doing in the cerebellum? And in the lab, we have a way of figuring this out. Before we get to that, asparagine is highly expressed in specific cerebellar cells called Purkinje cells. Okay, these are the largest neurons in the body, these triangular neurons. PCP2, so here's a red protein, a protein that's labeled with red color, and that shows where the Purkinje cells are and their morphology. So these are Purkinje cells, and they're triangular. They're huge cells, huge, massive neurons, and they're dendritic processes going into the molecular layer of the cerebellum. We don't have to know that. And green is PTPRD, the asparagine receptor. And how we figure out whether a protein is present in that cell or not is through a type of study called colocalization. We look for the two of them overlapping, basically. So when you overlap green with red, you get yellow, and we get lots and lots of yellow. So this is pretty good proof, and we have other ways of showing it too, that PTPRD is highly expressed in Purkinje cells. When you get rid of PTPRD, you knock it out, red remains red. It doesn't become yellow. Okay, so that's... Now, Purkinje cells had been studied, might have been the first neuron in the body to be studied. I could be wrong about that, but they're big, so they're easier to study, and their function is balance and coordination. That's really the function of the cerebellum, balance and coordination. What is the asparagine receptor doing in a cell that controls balance and coordination? All right, so in the beginning, the hypothesis was it's controlling appetite, not only through the hypothalamus, but also through Purkinje cells. just a hypothesis. Turns out that hypothesis is wrong. And I'll show you the data in a second. And how you go about answering that, and because all we knew was glucose and appetite. So naturally, we didn't have any other hypothesis. And we knew that the appetite was coming via the brain and the glucose was coming via the liver. So appetite. And it's not far-fetched. Two neuronal types, even far away, often work together. These dendritic processes and axons are very long. So it wasn't far-fetched at all that Purkinje cells make contact with AGRP neurons, and the two neuron types work in concert with each other to coordinate appetite, to control appetite. I jumped ahead. So not only is asparagine present in the Purkinje cells, it activates them, same as it does AGRP neurons, which I showed you. But here's a different technique. Basically, this red stuff is Purkinje neuron activation, and you throw asparagine into the brain with an ICV injection, intracerebroventricular injection with a spinal tap, right? And these neurons become active. When PTPRD is not there, so that's this blue line here, you can't even see it. These neurons no longer respond to asparagine. So pretty good proof that this receptor, asparagine receptor, is necessary for asparagine-mediated activation of Purkinje cells. But what does it do there? And I already mentioned that, our first hypothesis was food intake. So when you measure food intake in those mice where the asparagine receptor has been deleted, specifically in Purkinje cells, there is no impact at all on food intake. And of course, we kind of knew that we'd find this because when the mice were born and they got older, there was no change in body weight, no difference in body weight. And I've just shown you two examples. You get rid of PTPRD from all cells in the body. I showed you that knockout mouse, remember? The one that was made in Japan, and those mice weigh less and eat less. But not the mice where you delete this receptor, same protein, only in Purkinje cells. But luckily, the way we were measuring food intake was using something called a Promethean metabolic chamber that also measures other things. So here's another piece of luck. We found that the mice drank less water. This wasn't part of our hypothesis. We had no idea whether thirst wasn't on our radar. Why would it be? But the fact that this machine was there and it was measuring a bunch of things, water intake and food intake, and we were using it just for food intake, we got lucky. We found that there was a reduction in water intake, even though there was no reduction at all in food intake or body weight. So this put thirst on our radar for the first time, okay? In contrast, when we remove the receptor just from AGRP neurons, the neurons in the hypothalamus that control food intake, you get a reduction in food intake and body weight, but no change in thirst, no change in water intake at all. So this is a very clear example of the same protein, same receptor, deleted in two different types of neurons, producing two completely different physiologies. And they do not overlap. One cell type in the hypothalamus, the food intake center, affects food intake. And in Purkinje cells, which are not associated with thirst in any way, shape, or form, except a couple human studies where, 20-year-old studies, fMRI studies, where folks were made thirsty on purpose, and you looked at blood flow in the brain, the cerebellum kind of lit up like a Christmas tree. It's a correlative study, but telling those experimenters that, water intake, that might be something that the cerebellum also does. But nobody knew any more than that. There was this correlative human study, but that's it. This was the first example of you messing with the Purkinje cell in any way and getting a thirst phenotype, a clear thirst reduction. Okay, so this we published, basically came out in print like last month. And the title of this paper is The Cerebellum Modulates Thirst. Asparagine is not even in the title because no one knew that the cerebellum not only controls balance and coordination, it also controls thirst. Now this is fact, and it's out there. Surprisingly, when we measured balance and coordination from these mice that had knockout of the asparagine receptor from Purkinje cells, there was no change. And we measured it like 20 different ways. In all kinds of challenging tasks, there was no change in balance and coordination. So what that tells us is the same neurons can control two very divergent physiologies, balance and coordination and thirst, without any overlap. So this signaling mechanism, asparagine, its receptor, Purkinje cells, controls only thirst, has no impact whatsoever on balance and coordination. And however balance and coordination is regulated, doesn't appear to have any impact on thirst. Same neuron though. Okay, it's doing two very different things. Okay, and I mentioned that I'm telling you a story, right? So this story began way back, and I mentioned a discovery that just came out last month. Now, it's a super exciting time in the lab because ever since we discovered PTBRD, the asparagine receptor, we know all the places in the brain that it exists. And we can systematically knock it out and ask the question, what else does asparagine do? That wasn't available to me from information that the patients gave me, and is not just readily visible on the mouse surface or the human surface. Does that make sense? So now we're on the verge of major new functions of asparagine and its receptor, and it's involved in a bunch of like central things in the brain. So memory, anxiety, pain. We're developing therapeutics for all these. But asparagine is a very strange hormone. I got a ton of flack after that first paper came out, okay? And the flack basically came from the fact that it's created from a large structural protein. And folks who studied that large structural protein, their main question to me, full of skepticism about our paper and data, was, hey, how does this make any sense? How can a hormone, and hormones generally pulsatile, they go up and down, they do their job, and then they disappear, okay? And asparagine is fairly pulsatile too. It's circadian in nature. It goes up with fasting, goes down when you eat. It does all that. Like, what sense does it make for a hormone that's controlled like that, like all other hormones, to be attached and come from a large protein that is fairly stable in the body? It's a structural protein. I've taken the tack of like answering that question with like, I don't know, and it's not my job. Nature created the system, and what the heck do I know? Like why it created the system. But it is real because we found all this, and we published a bunch of papers on it, and every which way in which we test, asparagine does various things. And of course, the only way it gets created in the body is by getting cleaved off of fibrillin, okay? But still, let's consider this question and consider the fact that it's a strange hormone to be doing this. So viewed through the lens of classical endocrinology or classical extracellular matrix biology, this makes no sense, or very little sense. Hormones are generally not created this way, except that they are. So asparagine is not alone. So when you start thinking this way, you're like, okay, I'm going to comb the literature, and Bijou, who's my MD PhD student, is the one that did this. Like, what else is made like asparagine from a large structural protein? So here's asparagine from fibrillin 1. After our paper, a different group discovered placensin. It's highly expressed in the placenta, not adipose tissue, so placensin, which comes from fibrillin 2. It's also a glucogenic hormone, okay? Phil Shearer's group at UT Southwest, Albert Einstein, back in the day, discovered endotrophin, which comes from collagen 6, another large structural protein. And Judah Folkman discovered that endostatin comes from collagen 18. All right, so this is not weird. I mean, it is weird, but nature is doing it. And there are more examples than one. And nothing happens in nature just one time. So the fact that there are other examples is something to be expected. If you're right, and all the data points towards it, this is the normal result. Okay, where's all this going? So we published this. And so once we recognize this pattern, we're like, okay, we need to tell others about this pattern. Nobody has recognized this pattern yet. And no better way of recognizing a pattern than giving it a name. So we call these hormones that are made from large structural proteins. I'll read a little bit of this abstract. Cautamins, cauda meaning tail. So members of this class are derived from a cleavage event that also generates a much larger functionally unrelated non-hormonal protein. All right, so that's the dogma. We proposed this dogma. It is undoubtedly real because it's already happening, right? We have examples of it already happening. But the main thing it does is you can go fishing for more. So we started with one patient, and now we have 50 additional cautamins in the lab, approximately 50. They're present in you and I. Okay, they're present in all of us, in our blood. We've looked. We don't know what they do. Could be any function under the sun. Asparagine's functions we discovered because the patients without it, this experiment of nature happened. Couldn't do certain things. Regulate their glucose, regulate their appetite, regulate their body weight. We don't have that luxury now. So we've had to design a strategy. And I won't get into what that strategy is, But we have identified 2 brand new hormones with functions in the brain and the muscle. This could be literally, they could have functions, anything under the sun. And we will unveil them over the next 2 years. But this is working. And again, I'll tie it back to the fact that all this began with one patient. Unprecedented. Human genetics is an unprecedented Pandora's box of discovery potential. One patient. Generally, like clinical studies, lots of clinicians here. Clinical studies, you need thousands or hundreds, at least hundreds of patients to make sense of the signal, separate the signal from the noise. Not in human genetics. If you have an extreme example, one patient is enough. Okay, this is a real pitch for human genetics, for medical genetics. Okay, I have a lot of people to thank, obviously. Patients, the story began with, my graduate students, MSTP students, they've done all this work, postdoctoral fellows. Illa now has her own lab at the University of Kentucky. Our lab is a rotating sort of door. Lots of undergraduate students come through there. Couldn't have done all this neuroscience without collaborators. We don't know how to measure a neuronal activity in our lab. But we don't have to learn because there are others who know how to do it, and we make friends with them and ask them to do it for us. And then they publish papers together. Good way to make friends too. Roy Silito and Aaron's lab, they helped us with the cerebellum studies. We didn't know anything about the cerebellum. We didn't really know anything about thirst. Those guys helped us figure out that the mice had no problems with balance and coordination. They had all the fancy equipment that was necessary to measure it. Academic funding sources, university hospitals, Harrington Discovery Institute. So I was recruited here in 2018 by Jonathan and Dan. And 2018, if you guys remember, was the year that LeBron was leaving Cleveland. So when the offer came through, I'm like, nah, I'm not going. I like to think that rational people would respond that way. But we had long conversations. Dan and Jonathan convinced me that, LeBron shouldn't be a part of my decision making. So we've been very happy here. It turned out to be a good decision, and we've been productive, and we couldn't have done it without UH and HDI support. Thank you.
Thank you so much, Dr. Chopra. Fantastic, fantastic talk. I actually have a book on my bookshelf that's only single patient studies. So starting, of course, with Banting and Best and going on from there. So I really appreciated that you highlighted that so much investigation starts with the patient in front of you. Questions? Yes. There's, oh.
Back there.
I'm pointing back there, but is there somebody over here who had the mic? Or oh.
Since you framed your talk in the form of a story, I was wondering if you could speak to any communication you've done back to the 2 individuals in Texas and how they've been either kept apprised or even helped from the research, even though it's still ongoing.
Right. So we've stayed in touch. Abby's story actually came out several years ago in the New York Times. The article is called The Thin Gene, and it's told from Abby's perspective. And both patients had been bullied on the basis of what they look like. And one of them, Lizzie, the reason I found her really is because she's a public figure as an anti-bullying advocate, right? You can see how that would make sense. After the first story came out, they both came over to Baylor, and we discussed the results and what it means. And for the first time, they had some inclination of why they didn't like food as much. Actually, they thought that they liked food a lot. And that's why you have to measure it. You can't just take a patient's word for it. Because they were eating more frequently, the first thing was a misdirection. Like the first thing that came out of Lizzie's mouth was, I eat 8,000 calories in a day. We're like, this has got to be uncoupling. This has got to be too much energy expenditure because that's the only way you can explain this. Well, when they went into the metabolic chamber and Nancy Butte measured them, that was false. Okay. They both have Marfan syndrome as well, and I didn't get into the genetics of how that happens. Fibrillin is messed up too. It's not just asparagine, so it's a complex phenotype. So I often get calls when they end up in the hospital for various Marfan-related things, and then I coordinate their care in Texas. They both still live in Texas. Had a patient come from Utah, same disorder, a little kid, 11 years old. I can't offer them. So what would the treatment be in this case? At least for the asprozin part, it would be giving them more asprozin. Here's the reality of drug development. I cannot develop a drug for 10 patients in the world. Under no economic model does that work. Why do I talk about the economic model? Because that's the only way drugs get developed. It takes 30 million to even get to humans, do all your IND enabling studies and get to phase 1. And investors are not in it for the love and goodness. They're in it to make a return. I cannot raise money for a disease with 10 patients. In fact, my last company, Aseragen, we had a disease with, we were working on a disease with like 1,000 patients, and that was hard. 10 patients, forget it. It's not going to happen. So we can make asprozin in the lab, and we can make it research grade, but there's no money. There's no way I can develop it into GMP grade, test it in a rodent model and a non-rodent model for safety, do a non-human primate study, file an IND, hire a regulatory expert to do that, get approved to do human studies, and then pay for a phase 1, phase 2, and a phase 3. Phase 2 even. Sometimes for orphan diseases like this, you can get by with just a phase 2. So that's the reality. I have not been able to develop a drug to help the patients directly. But asprozin has taught us a whole bunch about it's elevated in obesity, and we're developing it for that. And primary polydipsia we're thinking about because of this thirst, and a whole bunch of other drug development that's coming up based on the new things that we're learning, for which there appear to be many more patients. Does that answer your question?
Hopefully this is on. Yes. Thank you for your storytelling also, especially for the non-MD population of us. We very much appreciate that. But I was wondering, we're all PMs in research here in this row, and I wondered how you prioritize now which of the codulants you're going to experiment with first, since you have a plethora now.
The short answer is we don't prioritize. I'm a big believer in the fact that the universe opens up and allows you to make some discoveries, and doesn't open up and the door is closed for others. We will take what we can get, is the approach that we take in the lab. If manpower was unlimited, we would work on all 50 at the same time for high throughput. And I'd be telling you that, hey, we haven't just discovered 2, we have 10. I'd be very happy with 10. And I would care very little about missing the 40. And remember, this is an arbitrary kind of screen anyway. There was a threshold that was established. There are potentially many more than 50. We established some stringent thresholds, and 50 popped out from that. So I guess in my business, we will accept what the universe gives us and not worry so much about the ones that don't come up. But I can tell you this, because I think that's the motivation behind your question. How do you tell which one's important? They're all important. I think asprozin is a case study for how obscure things are important. If it's in the body and has been kept around for thousands of years because of evolution, it's important. And we figure it out. There'll be other diseases too. I could give you other examples of the 2 that we're working with. There are diseases that are already apparent from the 2 that we seem to have discovered. Right? They're all important.
Great, thank you so much. Really appreciate that patient-centered talk.